So that's hearing in a nutshell. And it depends upon the activity of the sensory hair cells, seen here in cross-section and drawn in to make them more visible, which communicate through chemical synapses with afferent nerve fibers that carry that information to the brain. So that's hearing. And we know that hearing is carried by what are called the type 1 afferents of the cochlea, which make up 95% of the nerve. So virtually all the nerve fibers carrying information to the brain are this class of type 1 afferents, which are shown here in blue, and which connect up with a selected class of hair cells within the cochlea, sensory hair cells called the inner hair cells. And everything we know about hearing can be explained by the activity of these type 1 afferents. So their activity patterns correspond to our sensitivity, our frequency selectivity, our dynamic range, our temporal coding. These are the neurons which are responsible for hearing as we understand it. There's a few other afferent neurons, though, in the cochlea about which we know relatively little. In fact, essentially nothing. About 5% of the afferent neurons that innervate the cochlea are small diameter axons. They're unmyelinated, and we don't know what they do. They have a very different innervation pattern. Instead of synapsing with these so-called inner hair cells, they project out to the cochlea where they propagate for some distance, hundreds of microns along the length of the epithelium, and contact 10 to 30 of these so-called outer hair cells. Now, they've been very difficult to study because they're so few in number, they're small, and anyone who does a study of hearing is basically going to miss recording from them if that's your strategy. So we took a slightly different approach in our laboratory, which was really inspired by our conversations with our colleagues who study pain in the central nerve, in the peripheral or somatic nervous system. And part of that inspiration is from the fact that there's a kind of analogy here between these large diameter myelinated axons that serve the cognitive aspects of this sensation of hearing versus small diameter unmyelinated afferents, which serve something else. And in the somatic nervous system, these are the C fibers that are responsible for carrying the information about pain rather than cognitive touch. So we saw an analogy here, and there were other kinds of clues which led us to say this might be important to look at these kinds of type 2 neurons. So to solve the technical problem of being able to record from them, a graduate student in the lab named Kat Weiss took the step of isolating a piece of the cochlea, shown here on the right of the slide. So this is the sensitive sensory epithelium from a rat's inner ear, which has been dissected free from the bone. She cuts a piece of it off and puts it into a dish. The tissue remains alive for long enough to do electrical recordings from the sensory hair cells or from the neurons that innervate those sensory hair cells. And so in doing that, Kat was able to apply an electrode to the type 2 afferent out here in the sensory epithelium near to where it makes its synaptic connections with the sensory hair cells. And from this kind of effort, it began to be possible to learn something about what these particular rare kinds of afferent fibers in the cochlea might be doing. Now, I won't tell you about all the details of those experiments. I'll just summarize what we've learned to date, which is that although these type 2 cochlear afferents connect up with a large number of sensory hair cells, in fact, they are very poorly driven by those hair cells. For an equivalent amount of excitation, the type 2 afferents respond very much less than the type 1 afferents, which are responsible for carrying information about sound. So the type 2 afferents are very insensitive, and sort of projecting that into the acoustic world, it would imply that they were only capable of responding to very loud, possibly traumatic sound. And interestingly, these type 2 afferents are not only sensitive to synaptic communication by the sensory hair cells, but they also are excited by adenosine triphosphate, ATP, which is a chemical that also mediates painful sensations in skin. So we think, again, the analogy is growing stronger by these studies that maybe these unusual rare afferents in the inner ear are responsible for mediating not hearing, but the sensation of traumatic or painful sounds. Do the type 2 afferents carry ear pain? Don't really know yet. There's much more work to do. But we think that there's going to be some interesting implications for thinking about pathologies of the inner ear as a consequence of studying this class of afferent neurons. So for example, I mentioned at the beginning that hyperacusis is a condition where loud sounds become more painful than ordinary. And so that is a situation which may have something to do with the fact that the type 2 afferents become more sensitive or constitute a larger amount of the information flowing to the brain. And one of the conditions under which that happens is after acoustic trauma, where the type 1 afferents are very sensitive to trauma. About half of them, in some cases, lose their connections to the sensory hair cells, and they fail any longer to carry information to the brain. The type 2 afferents are resistant to this kind of damage. After acoustic trauma, they're still there. They seem perfectly capable of responding, and so what now results is an imbalance in the information pathways going to the brain, whereby what may be the trauma detectors are gaining ascendance over the normal cognitive listening neurons. And so this may also have something to do with the kinds of conditions like tinnitus, which is also something that arises subsequent to damage in the auditory periphery. Finally, to bring the story full circle, taking our techniques for recording from small fibers and isolated bits of epithelium, now together with Michael Katerina and Jin Jiandong, as part of the pain work group, we're going to take those same kinds of methodologies and apply electrodes to small fibers and excised pieces of skin to try to understand the transduction mechanisms of pain in that setting, and to see again what sorts of parallels we may be able to draw with studies between the different sensory organs. A collaborative and cooperative study which has resulted directly from these Center for Sensory Biology and the pain work group and other such mechanisms at Hopkins. I'll stop there. Thank you very much.